This is chapter 4, growth factor signaling in oncogenes. And we're looking at growth signal autonomy of the cancer. And the one of the big thing we were talking about, the EGF pathway. EGF, of course, stands for epidermal growth factor. And EGFR, the EGF receptor, has other names like ERBB1, ERBB2, and HR1. And these receptors are receptor tyrosine kinase, and there's once EGF binds to EGFR, then they're like shape changes and they dimerize with each other, and then that allows them to autophosphorylate. And once that happens, then it brings in the GRB2, and GRB2 has the SH2 portion that binds to the phosphate in the EGFR, and then the other side of GRB2, which binds with SOS, it has the SH3 domain, and once the SOS is like bind, that helps the RAS with GDP to turn into RAS with GTP. And once this is with GTP, then it causes that RAS activate RAF, and RAF activates MEK, and the MEK phosphorylate MAPK, and MAPK finally phosphorylate transcription factor and allows more gene expression, hence causing more. So that's why it's proto-oncogene. Kinase is the adding a phosphate group, and phosphate is removing. And then the pathway that I just explained, where it goes from the RAS with GTP to all the way down to MAPK phosphorylation. The transcription factor is a serine threonine kinase cascade. So there's that. And what it also does is that the RAS, that one with the GTP, also activate the PI3K pathway. And how that works is PI3K converts PIP2 into PIP3, and the PIP3 binds with PIPK1, and then PDK1 phosphorylate AKT, and then it also activates transcription factors, and also mTOR, which synthesize lipid and nucleotide. And the SRC, which is the first oncogene to be found, interact with transmembrane cell sulfur receptor and mediate transduction of intracellular downstream signaling pathways. We have the PI3K pathway, we have RAS pathway, which is and then BRAF, MEK, ERK, and then transcription factor. Transcription, we have Rho pathway and then JAK pathway as well. And then the MIC transcription factor family is another important topic. So we have this MAX transcription factor, and if it binds with MIC, then it leads to cell division. On the other hand, if the MAX binds with MAD or MXI1, then it blocks the cell division. So MIC and MAX causes cell division, and MAD/MXI1 binding with MAX blocks the cell division. So mechanism that activate proto-oncogenes to become oncogenes, there's a point mutation/deletion in coding sequence that leads to structural and functional changes. Point mutation and deletion in regulatory sequences can lead to overexpression. We also have a chromosomal translocation, which is just like fusing different pieces of chromosome together, which fuse protein with novel functions, leading to some potential mutations. Insertion mutagenesis caused by viral integration. virus inject their DNA into human DNA for them to basically reproduce themselves, but they can also lead to abnormal expression. And we also have gene amplification, which increase gene doses and protect production and protein production. And that translates. We also have the ecDNA, which is extra chromosomal DNA, and 1/4 of the solid tumor has related to ecDNA. So in this example, we see that their ecDNAs, extra chromosomal DNAs, are unevenly divided during cell division, and so some cells has more copies than others. Um, because they are not part of the chromosome, so they can be spreaded unevenly. And the significance of ecDNA is that unlike linear chromosomal DNA, which has a higher order compaction, because of its structure, it does not compact as low as much, and leads to it's more likely leads to more expression or a higher chance of activation of the oncogene, which leads to cancer. Um, and people studied oncogenes and their mutation through cell transformation assay and Maxim-Gilbert sequencing. Cell transformation is so you put tumor DNA, you get tumor DNA from tumor, and then you mix it with calcium phosphate and then transfect it into human cells. Or in the case of Maxim-Gilbert sequencing, you mark it with a radioactive marker, and then you cut it with enzyme and chemicals and do gel electrophoresis. In terms of therapeutic strategies, we can either do a monoclonal antibodies. So they're just they're only targeting one specific site. That's what that means. And so we have this different drugs for monoclonal antibodies, Herceptin, Erbitux, and Vectibix. And then we have the EGFR targeting, where it has Arisa, Gurtif, and Tagrisso, and stuff like that. You can also target RAF, MEK, or yeah.